Namaskar दोस्तों, welcome to channel Toxicology on board. Today in this video we'll be discussing another DABT topic that is ICH-S8 guideline, immunotoxicity studies for human pharmaceuticals. So from the recertification question papers, I didn't find much questions on ICH-S8 guideline. Recently, if you see past 5 years, most questions they are asking on ICH safety guidelines as well as quality guidelines. So uh, from the last previous question papers, you couldn't find much questions on safety or quality, but the only those people who have written the exam in 24 to 2025, they can only suggest us what are the questions nowadays examiner is asking with respect to safety guidelines. So I have taken questions from ChatGPT. So 5 questions I'll discuss in this video. We'll try to cover to understand the different aspects of the guideline. Eventually, in next subsequent videos, each and every sections from the guideline we'll discuss. Okay. So in this we'll discuss only question with respect to what the guideline, important sections in the guideline. Okay. So what is the primary objective of ICH S8 guideline? So whenever we are reading any safety guideline, whether it is S1, S2, S3, whatever the quality guidelines also or multidisciplinary guidelines also, one question examiner will ask you with respect to objective or scope. This guideline is applicable to what type of pharmaceutical or what type of chemicals and what type of chemicals or pharmaceuticals are excluded out of scope or out of objective. So this question will definitely come. So let's understand in future some other time we'll try to only make one video only on objectives like S1 what is objective, S2 objective like that. Okay. So, let's stick to S8 guideline here. So objective of this S8 guideline is right answer is in the green text to identify the potential adverse effects of pharmaceuticals on the immune system. For example, you are developing a certain conventional molecule. See, this guideline, ICH S8 guideline, is not applicable to biologics. We are talking only about small molecules, that is traditional molecules, conventional molecules. So, if you are developing a certain anti-lipidemic drug or antihypertensive drug, whether my investigational drug, whether it is going to affect the immune system, is it going to either tweak the immune cells, that is T receptor cells, T helper cells, these cells are there, no? It should not trigger the immune system. It should not cause either immuno enhancement or either it should not cause immunosuppression. Okay, these or immunotoxicity, it should not cause. So that is a major thing. So whether this to identify the potential adverse effects of pharmaceuticals on the immune system. Okay, so these are on the right side. You can see these are the different immune organs which in our body which are responsible for the immune system. Okay, so let's see what are the different options are there which are not applicable or not right for this particular ICH-S8 objective. First option is to establish the efficacy of this immunosuppressive drugs. So pause here, try to understand when you will take the immunosuppressive drugs when a patient is undergoing transplant, organ transplant is there. There you want your organ not to whatever the newly transplanted organ is there, no, it should not reject your body. In such a case, what you will be doing, the physician will be prescribing you. Additionally, after the surgery, he will be prescribing you to take immunosuppressive drugs. So they are different type of category of drugs. So these immunosuppressive drugs will not fall under ICH S8 guideline. Okay. So to establish the efficacy of immunosuppressive drugs, no. The efficacy के लिए again you have to what type of studies means ICH S4 guideline or efficacy study, whatever are models are there that you have to do. S8 is not applicable for immunosuppressive drugs. If the intended effect is suppression of the immune system, no. Whatever it should be unintendedly, it should affect the immune system, either immunoenhancement or immunosuppressant, like that. Okay, little bit confusion. Try to understand because you have to read immunotoxicity. You have to also read immunogenicity. What is the difference? Also, one video I'll try to make in next upcoming videos. Next, third option we'll see to replace all standard toxicity studies with immunotoxicity studies. This is absolutely wrong because we have to follow 3R principle. The main agenda of this all these new ICH guidelines is application of this 3R principle. So in that, in addition to the standard toxicology studies, what we do, for example, repeat those 28-day study, 90-day study, 1 year, 2 year, or segment 1, segment 2, segment 3, reproductive studies, carcinogenicity studies, and genotoxic study, which are the battery of toxicology studies. In addition to that, you also have to check for whether my new drug is going to affect the immune system. So, to replace all standard toxicity studies with immunotoxicity studies, no. Immunotoxicity study will be add-on to the existing existing that battery of toxicology studies. So that you have to remember. Okay, why? Because we have to Apply 3R principle. Last one is to evaluate only vaccine-induced immune responses. So for vaccines as well as for biologics, we have separate guideline. For example, for biologics, you have ICH S6 guideline. So this is out of scope of this particular ICH S8 guideline. Okay. So what is the explanation which is given by the ChatGPT? We'll see. ICH S8 provides guidance for evaluating the potential of the human pharmaceuticals to cause adverse effects on the immune system. Okay, I am developing a new drug. If my new drug is going to affect the immune system, so that you have to check. Okay, unintended immunotoxicity assessment is a integrated into overall non-clinical safety evaluation. So this is your option number C. Okay, you have to not replace. It has to be integrated. So along with the battery of toxicology test plus add immunotoxicity, or you can include the immunotoxicity, whatever the endpoints are there, to the same 28-day study or 90-day study or 6-month or 9-month study, you can add it, repeat those toxicology study. So let's see what is the guideline it is saying. That is section 1, ICHS guideline, section 1, that is introduction, objectives of the guideline. We'll quickly read. See, the objective of this guideline are to provide recommendations of non-clinical testing approaches to identify compounds which have a potential to be immunotoxic. If I'm developing a new drug and if it is showing certain red flags, it is showing certain indications that yes, this particular developing drug might trigger the immune system, then you have to, this particular guideline is going to give you an idea what kind of additional immunotoxic studies you can do. Next one is, second one is um, guidance on the weight of evidence decision making approaches for immunotoxicity testing. Okay, so as we told now, we have to follow the 3R principle. We have to apply integrated assessment, that is weight of evidence approach. So from the already existing repeat dose toxicology study, gene tox studies, from that only, if we can look into leukocyte count, if you can look into helper cell counter, like this, from the existing essays only, we can look into the smaller details from that evidence. You can integrately, you can think and you can plan whether I should do an additional study or with the existing information, uh, this molecule appear to be safe or you can mitigate the risk. Okay, so the objective is guidance on weight of evidence decision making approach for immunotoxicity testing. Immunotoxicity is for the purpose of the guideline defined as unintended immunosuppression and immunoenhancement. So this is very, very thing that you have to keep it in your mind. Unintended. Intentional is immunosuppressant drugs. But the newly developed drug, if it is triggering, it is causing immunotoxicity or immunoenhancement, it should be unintended. Okay, drug-induced hypersensitivity. There are certain drugs, for example, penicillins or beta-lactam antibiotics, which themselves trigger the hypersensitivity reaction. Okay, they might be triggering IgE, IgM, four types of hypersensitivity reactions are there. No, I have one video on the types of hypersensitivity also. I'll give the description in the link. So, and autoimmunity are so there are certain drugs which themselves will trigger autoimmunity. So those things are also excluded from this ICH S8 guideline. Pause the video. I would request you go and check what are the drugs which are, for example, I told you about beta-lactam antibiotics and penicillin. So similarly, you make a list of autoimmune drugs. What are the drugs which are going to cause autoimmunity? Okay. So like that you can remember. So excluded things are drug-induced hypersensitivity and as well as autoimmunity. So that was about the first question that we discussed about the objective scope. What does this ICHS guideline covers? Now we'll see second question. Second question is which approach is recommended by ICHS8 for the evaluating immunotoxicity potential of a pharmaceutical. Now at this stage we have reached now we have realized that whatever the new molecule which we are developing, it is going to affect the immune toxicity. It is having adverse effect on the immune system. Now, what are the next step as a researcher, as a pharmacologist, as a toxicologist? What additional things that you can do for your company that we'll see? Okay. So to in order to safeguard the molecule or in order to further develop your newly investigation molecule. So first one is so right answer is what is the approach that you have to follow is weight of evidence approach based on relevant scientific information. As I told you, from the battery of toxicology test, you have to see small indications, whether it is affecting my T helper cells, whether it is affecting if there is any leukocyte count, granulocyte count, those things, whichever the immune system organ weight change. So these things, what we will see in section 3.2, we'll see what are the selection of the essays from the battery of test from which we can get the same information. So weight of evidence approach based on relevant scientific information. This is the approach that you have to ICH S8 is recommending whenever you are finding that evaluating the immunotoxicity potential of a pharmaceutical. So what are the other options we'll see which are not applicable? Routine testing of every immune function using all available assay. new type of assay, it is not at all recommending ICH S8 guideline. So routine testing of every immune system, like whether it is affecting your thymus, whether it is affecting your thyroid, whether it is affecting your endocrine gland, all those things of additional new type of assays are not needed at this particular point of time. Testing only in humans before approval. This is also very wrong. Before initiating phase 1 or phase 2, already this immunotoxicity related data should be available and you have to submit to the regulatory agency. Then only they will give you a green signal to go for phase 1 trial. Okay. Next one is performing only a single in vitro assay. Always remember, whenever we are able to establish some certain toxicity of a certain chemical, when you are giving that this particular drug is going to cause some potential adverse effects, you're not going to give it with a single essay. You have to have a battery of essay or confirmation essay should be there. So that certain times there will be false positive or false negative from this one part if you are relying only on one essay. That too, it is an in vitro essay. If there is an in vivo model, you have to try to prove it with the in vivo model also the same positive finding. Okay, and it's also case by case. You cannot just say on this thing. So the, for that reason, this option is also not applicable. This approach is not applicable for ICH-S8 guideline at this point of development of the molecule. Okay, so this is the weight of evidence approach. You can see what is the explanation. We will see. ICH-S8 recommends a weight of evidence approach. Information from pharmacology, that is from the EC50 or whatever the models are there, no, the pharmacology models for which you are developing the drug. That from the data you can get it. General toxicity studies, that is your 28-day or LD50 acute study if you have done, or if you have done some irritation study, or you have done some 90-day study or 6-month study. Clinical data. If you have some information on the clinical data for the same class of molecules, drug class as a discussed mechanism of action also. What is the target receptor this particular drug is going and affecting, and other relevant sources is considered to determine whether additional immunotoxicity studies are needed. Whether we should do for other additional studies or not, how we are going to by considering all these approaches. So that is called weight of evidence approach. So standard toxicity studies, pharmacological properties, structural similarities, drug disposition, where the drug is after you take the drug, where in which part of the organ, whether it is going to kidney, liver, or tissue, where it is going and depositing, intended patient population, is this drug recommended for only female population or male population, geriatric population, or children population, that you have to see, and clinical information. If there are some trials are there, similar class of drug, from that also you can, from those articles also you can get the information. So once you have performed this weight of evidence, no cause for concern. If you find that in from this studies there is certain trigger, certain red flag is there. Yes, it is going to affect the thyroid organ, or it is going to decrease the weight of this thymus, or something like that, or there is an elevation of this granulocyte, white blood cells, or TL cells like that. So if there is a cause for concern, then you have to go for additional immunotoxicity studies. And what the ICH-S8 guideline recommends is two assays are there. One is tier 1, that is TDAR, T cell dependent antibody response, and second one is immunophenotyping. So these two things we'll discuss in next videos also. Okay, what are those assays? TDAR assay, what it is, and what is immunophenotyping. And if there is, if you found, or from this weight of evidence, you found that it is not going to trigger any of the even potential, there is no potential concern, then no additional immunotoxicity studies are required. Okay, this is the recommendation guidance given by the ICH-S8 guideline. So for this, no, to understanding weight of evidence approach, you have to read from the guideline section number 3.2, that is selection of the assays. So if the weight of evidence review indicates that additional immunotoxicity studies are called for, there are a number of assays which can be used. If there are changes in the standard toxicity testing data, as I told you, if there is a change in organ weight or the or T cell count or like that, a number of assays which can be used if there are changes in the standard toxicity testing data. Suggesting immunotoxicity. The type of additional immunotesting that is considered appropriate will depend on the nature of the immunological changes observed and the concerns raised by the class of the compound. So if this is anti-hypertensive drug or anti-lipidemic drug or anti-diabetic drug, based on that, it is recommended that an immune function study is conducted, such as T cell dependent antibody response, that is TDAR assay. If the specific cell types that are affected in standard toxicity studies are not known to participate in the TDAR assay, the measure function of the specific affected cell type must be conducted. So we have to read in this guideline. Additionally, appendix is there, that also we have to read, whereas specific target is not identified. So from the existing assays, if you are not able to identify whether it is affecting helper cells or WBC or RBC like that, Okay, and immune function studies such as TDR is recommended. In addition, immunophenotyping of leukocyte population. So you have to count even leukocyte count also you have to do. A non-functional assay can be conducted to identify the specific cell population affecting and might provide useful clinical biomarkers. So from these toxicology studies, you can identify that these are the biomarkers. Leukocyte count is increasing or granulocyte counting is increasing. So like that you can identify so that whenever you're initiating phase 1 trial or phase 2 trial, you can consider this as a biomarkers. So whenever you're giving it to this particular investigational drug to the patient, you can look for these kind of endpoints in that particular after administering the drug to the patients. Okay, so that was regarding about integrated approach, weight of evidence approach, how the existing information is going to decide whether further testing is needed or not. Okay, so next we will see third question. Third question is something related to similarly, what are the different additional toxicology immuno assessment is required or not? So which of the following findings from a repeat dose toxicity study may indicate a potential need for additional immunotoxicity assessment? So already repeat dose toxicology studies you have done, 28-day or 3-month or 9-month study you have done. So from the information from this study, how it is going to help you to decide? Let's see. Increased immune organ weight and histopathological changes. The right answer is if there is a change in the organ weight, particularly thymus or thyroid, like that, immune endocrine organs are there, no? And is there any change in the histopathological findings? If you find there is a change in the histopathological findings, then that will be your decision maker. What are the other options? See, normal food consumption, stable body temperature, normal vehicle control. So all these three are not triggering any red flag with respect to immunotoxicity. Okay, so we'll see what is the explanation which is given by the ChatGPT. Findings such as changes in the immune system organs, altered leukocyte parameters. I just told you, you can pause the video and check what is this leukocytopenia, leukocytosis, granulocytosis, lymphopenia, lymphocytosis. Okay, histopathological changes in the lymphoid tissue may indicate a potential immunotoxicity and can support the need for further targeted investigation. So if we find that it is going to affect leukocytes and if it is going to affect granulocytes, then our biomarkers, whatever will be there, will be focused towards targeted investigation will be there towards this particular type of cells and tissues. Okay. So for this to understand more in detail, you have to read ICH-S8 guideline section 2.1.1, that is standard toxicity studies. So what is this? Will very quickly will run through data from this STS, that is STS is standard toxicity studies, should be evaluated for signs of immunotoxicity potential. So what are the things that we have to look in these studies is signs that should be taken into considerations are following. Hematological changes. If there is any change in the RBC, whatever the blood profile, what you are doing, RBC, WBC, platelets, such as leukocytopenia and leukocytosis, granulocytopenia and granulocytosis, and lymphopenia and lymphocytopenia, cytosis. So these things you have to look in the RBC whenever you're doing blood, complete blood profile, CBC. Okay. Next one is alterations in the immune system organ weight. So our option was organ weight. So decrease here, what has happened? Increase in the organ weight. So certain times, so body will also undergo adaptive changes. If the new chemical we are xenobiotic, if you are taking, certain times, to meet the demand of that organ, to meet the increase in demand of the enzymes or the production, no, the organ size will also increase. So adaptive changes it is called. Okay. So in this case, there is an increase in the organ weight, alterations in the immune system, organ weight, and histology. So we have histopathology is also given in the option, and changes in the thymus, spleen, lymph nodes, and bone marrow. These are the your immune organs which are involved, immune system organs which are involved. Okay. Then we have other options like 3, 4, and 5 which are listed in the SAID guideline. Changes in the serum globulins that occur without a plausible explanation, such as effects on the liver and the kidney, can be indication that there is a change in the serum immunoglobulins. So we have to look for this also serum. If there is any alterations in the serum immunoglobulins, increased incidence of infections, are there any particular, since you are triggering the immune system, is are the particular test species, it is showing certain infections or not, that also you have to see. Increased occurrence of the tumors can be viewed as a sign of immunosuppression. Okay, in the absence of other plausible causes such as genotoxicity, hormonal effects, and liver enzyme induction. So whatever the test species, is it showing any development of the tumors? And the cause for those tumors should be related to this test item rather than genotoxicity, hormonal effects, or enzyme induction. Okay, so pause the video, read section 2.1.1, or you can go back and read in the guideline. So now we'll see another question. Question number 4, with respect to which factor is most important when deciding whether additional immunotoxicity studies are necessary. So you can understand why this is, because main purpose of S8 guideline, main objective is there any adverse effects on the immune system? If yes, what studies are there? That only it is the main objective of S8 guideline. Okay, so that's why you have to keep coming across the same objective like weight evidence, integrated assessment, what are the biomarkers, what are the triggering points. Okay, so the right answer will be the mechanism of action and pharmacological properties of the pharmaceutical. So whatever the new investigational drug which you are developing, you have to know the mechanism of action and you have to understand the pharmacological properties. So this is an important factor whether further additional immunotoxicity studies are necessary or not to identify. So what are the other options we'll see? The color and the physical appearance of the drug product, size of the clinical trial site, okay, packaging material alone. So these are all unapplicable in the case of the question, additional immunotoxicity, whether to for the decision making. So right answer, what is the explanation is the mechanism of action, pharmacological class, intended patient population. As it were, whether this we have seen in that particular section also. First year, second slide, we have seen duration of the treatment, findings from the existing non-clinical, findings from the existing clinical studies are important considerations in determining the need for additional immunotoxicity studies. So these are the uh, objectives to decide whether we have to go or whether we have to stop particular study. Okay, this is called integrated risk assessment. Okay, now we'll see question number five. Question number five is something related to overall safety assessment, the relationship between. Okay, which statement best describes the relationship between immunotoxicity studies and the overall safety assessment? See, finally, what is the main objective of any researcher, whether it is pharmacologist or toxicologist, is safety of the whatever the drug which you are developing. The drug should be efficacious and it should be safety. It should not cause any adverse effects on the intended patient. Okay. So right answer will be immunotoxicity findings should be interpreted together with other non-clinical and clinical safety data. With that we have seen from the battery of toxicology studies as well as same similar class. If there are some clinical trials are done, then that also you can consider and then you have to make the decision. Okay. What are the other options we will see? Immunotoxicity studies are completely independent of other toxicology studies. This is absolutely wrong. Immunotoxicity studies are repeat dose toxicity studies. No, only positive immuno findings are relevant for risk assessment. We have seen that it should not cause immuno enhancement and it should not cause immunotoxicity. Two things you have to see. Double-edged swords you have to understand. So ICH emphasizes an integrated assessment. Immunotoxicity findings should be interpreted in context of general toxicology, pharmacology, toxicokinetics, additional clinical information to determine is there any potential risk to the humans. Okay, that is the thing. You can pause the video and you can go through the picture here, when to stop the study and when to continue the additional studies. Okay, so this is the integrated slide from the ChatGPT only which I got with respect to how to understand the S8 guideline. So you can pause and look into. I will upload the same thing in the post also. So you can read how can we interpret the risk assessment, how the documentation or the what are the factors and what are the different tires approaches are there. Okay, so in next videos with respect to ICH guideline, in detail we'll discuss everything. We'll discuss TDRSA, we'll discuss immunophenotyping. Okay. Um, Thank you so much. Be curious. All the best for your exam.